I want to draw our focus down a little bit now, because all those changes in the Cambrian, I talked about the massive amounts of information that had to be added to get all those different basic body plans to suddenly emerge. Now we want to show you how what we're discovering now about how that information is organized. The whole idea behind what a gene is, it used to be one gene codes for one protein. In the early 90s, we began to see more clearly that that is far too simple a picture of what a gene is. We used to think of a gene as having a little, what was called, if you have a string of DNA here, and over on this side you've got what's called the promoter region, which is where something attaches, and that starts the messenger RNA being transcribed, and then that goes out into the out of the nucleus and into the cell, and that's where the protein is made from the messenger RNA. You had one gene, one messenger RNA makes one protein. This is a modern gene. This is what we see as a gene today. It's hard to know even where to begin. These exons, exon A, and there's two different versions of exon A, exon A1, exon A2. Exon B1, exon B2. Some proteins would be made with exon A1 and exon B1. Some proteins would be made with exon A1 and exon B2. Or exon A2 and exon B1. Always an A and a B, but it would shift whether it was B1 or A1. Other types of different types of modifying sequences, virus-like elements, silencers, tissue-specific enhancers. In other words, if this was a gene coding for proteins that are used in both kidneys and liver, well, this might enhance the gene in the liver, and this one would enhance it in the kidney. But it's all part of one sequence. And all your chromosomes have that same sequence. It just depends on what surrounding tissue experience it finds itself in, is how it's expressed. The imprinting element, those yellow segments over there, those have something to do with whether this chromosome came originally from the mother or from the father. All of your genes are imprinted as to whether they came from male or female. And it's interesting, early in embryological development, for certain developmental pathways, only the father's genes are used, and for other developmental pathways, only the mother's genes are used. We don't know why. It's doing it. And there's some organizational structure in the cell that somehow, because it happens the same way in all embryos. It's not just willy-nilly. It's somehow being controlled above. So this has left people, I saw Rick Sternberg, who was just in that previous video, give a presentation. I borrowed the slide from these next several slides from that presentation. The title of it was The Immaterial Gene, because we don't really know how to define a gene anymore. There are far too many variables, far too many complexities to the whole thing. Gene clusters we have now found. Genes, they're not just located randomly throughout the chromosomes. We find them clustered together on small portions of the chromosome. So we might have 5 genes for 5 different proteins that are involved in one particular biochemical sequence. But they all tend to be clustered in one small location on the chromosome. You've got gene 1, gene 2. Look, there's since DNA is a double helix, there's 2 strands, right? One goes this way and the other one goes this way. Well, sometimes the gene is coded for on different strands of the DNA. Here you see the arrow has, these are on the strand that's going this way, and gene 3 is on the strand going the opposite direction. And what we're seeing is you see some overlap there, don't you? We do find genes now that overlap. In fact, we're finding it far more frequently than anybody ever expected. I first heard about overlapping genes when I was an undergraduate student at the University of Illinois in about 1975. And it was only in a small virus. The virus genome was only about 10 genes to start with, and they found 2 genes that were overlapping. And they were really confused by that. And I thought, that's just really weird. It's just some goofy virus. How could that have happened? We're not going to bother with it. It's just one virus. It's an anomaly. No big deal. Now we're finding overlapping genes everywhere in the genome. Genes are clustered together. The protein coding part of your genome, remember, is only 2%. But for some reason, they're all shoved together in certain locations. They're not just spread out all throughout the whole genome. They're clustered together. And on top of that, the process for making a protein, you start with the DNA, you transcribe it to RNA, and the RNA is translated to protein. So the first step from the gene is to make RNA transcripts. These small blue arrows illustrate how many different transcripts can come from this one cluster of just 5 genes. Now, some of these transcripts involve more than one gene. Some of them start and stop in the middle of genes. They don't just code for, you don't just see a transcript for just one gene. They're all over the place, and we're not sure why that is. What the cell then does, it takes some of these transcripts and it cuts the transcripts up even further, and arranges those sometimes into new transcripts that make a gene, if you will, that will make a protein that you can't find a linear corresponding sequence in the DNA of the chromosome. Because it's coming from different places. It's coming from different transcripts, from different places in the chromosome, and sometimes from even different chromosomes. So what are we learning? The physical description of a gene is currently lacking. We don't have one. What we do know is that there's a hierarchical organization to how a gene is structured, how it's put together. There are multi-levels of optimization or of organization and control. There's many different types of information, and it's connected by what are called coding chains. Okay? There's adjacent genes. There's genes on other chromosomes that are connected in ways we never would have thought were possible in the genome. I got this paper from Rick Sternberg, and I saw him use this quote, and I've got to have that quote. It says, "The existence alone of overlapping protein coding frames is, quote, virtually impossible by chance." I wanted to get the paper because I want to see what do they do with that statement. This statement is the title of a section in the paper. And nowhere in that section do they try to explain what they meant. They use it as a title, and they never talk about it again. Now, I don't know how to explain that. I was really disappointed. Please talk about this some way, somehow. Tell me what you mean by that. No, they're not going to touch it. Another clue here. We can get, here's a gene, several different genes. You can get, he says, sometimes as many as 38,000 messenger RNA transcripts from a single DNA strand. DNA, what we would have called a gene. 38,000 potential proteins from one gene sequence. As I said earlier, what happens is that even within these RNA sequences, the cell somehow has another machinery involved that splices and cuts and dices these transcripts and reassigns them back together in different forms and different stretches. We don't even know what all the machinery is that does this. We don't know why it does this. And even at that stage, then you have to try to ask yourself the question, how did this evolve? I first learned about what's called RNA splicing here when I was a graduate student at UTD. We had found a small microorganism, a multicellular organism, that was doing this in its mitochondria, and we couldn't figure out why. They had long stretches of a single nucleotide, and these were uracils, so they abbreviated U. We had long stretches of U's in the middle of these genes. And what happened, as soon as the transcript was made, the U's were cut out. Nobody ever seen that before. And I remember asking the professor during one of the, somebody was presenting this as a journal club paper, and I remember asking the guy who was the mitochondria specialist there, I said, now, Now, I can understand how selection with this system in place, natural selection would maintain that because it's needed for the organism. But what's the selective advantage for this evolving in the first place? Kind of looked at me. Well, I don't know. Yeah, I don't know either. I'm waiting for an answer to that. So there are many, many functional sequences that can come even from these. Sometimes these RNAs are clipped apart into very, very small RNAs. And all of these very, very small RNAs all seem to have a function. They're doing something in the cell. But you know what? We don't have a clue what they're doing. We don't know what these RNA pieces are doing. But we do know they have an effect, because if you just cut them out altogether so that the cell can't make them, the cell dies. They're doing something. They're needed for something. They're not coding for proteins. They're regulating something. But we really don't know. Now I'm going to bring you back up to a macro level again. I'm going to bring the plane in for a landing here. This is a pileated woodpecker. I should have asked if anybody knew what it was. Got to be a naturalist, a bird watcher in here somewhere. Right? Woodpeckers. You ever think about woodpeckers much? They are weird. What do they do all day? Bang their heads in the trees, man. That's just really strange. Did you ever listen to a red-headed woodpecker or a flicker that knows how to do it really fast? I mean, we call it drumming because it sounds like a drummer's drum roll. It is so rapid. That's too slow. It's just really, really, really fast. Okay? Now, woodpeckers have a number of really cool adaptations. I'm a critter guy, okay? I'm a zoologist. I went to zoos as a kid, so I love the critters. But number of adaptations that allow them to do that, to bang their heads in the trees. If you look at the tail is buttressed up against the trunk of the tree. It acts as the third leg of a tripod for stability. The toes of a woodpecker. Most birds have 3 toes in the front, 1 toe in the back, especially for perching birds, to perch on a limb or a twig. The woodpecker has 2 toes in the front, 2 toes in the back to grasp the vertical surface of the tree. 3 and 1 is not going to work real well. 2 and 2, that's great. That works fine. Okay? The beak of the woodpecker is extremely hard. It's not brittle, it won't break. It's not rubbery, it doesn't bend. It has to hold up to the constant pounding. And there's actually a shock absorber between the skull and the back of the bill, so the bird doesn't crack its skull as it drums into the tree. Shock absorber is made of cartilage and muscle. Now think about the muscles in the back of the neck and the shoulders, the nerve endings. Now think about, I saw a few of you trying to do it, you know. Now our younger headbangers have learned that you do too much of that and it's not good for your head. Our heads are not designed, if you will, for that rapid back and forth movement. Woodpeckers do it just fine. They do it all day. So they've got a unique set of muscles, nerves, bone structure in their skull, neck, shoulders to allow them to have that rapid movement back and forth and the pounding it takes. But that's not the most unusual thing about woodpeckers. Why do woodpeckers bang their heads in the trees anyway? What are they doing? They like headaches? Insects. Where are the insects? In the tree? Sometimes, like for the pileated, what he does is he just kind of flicks the bark off and he eats the insects that are just underneath the bark. But a lot of them, the other woodpeckers, are literally drilling holes into the tree so they can gain access to the insect larvae that have created tunnels in the wood of the tree, and they're drilling to get to those tunnels, and then they insert their tongue into the tunnel and search up and down for insects, insect larvae. Now they have a very sticky saliva on the tongue. They also have some little barbs on the tip of the tongue that when they get one, they can be successful in pulling it out. But did you hear what I said? They stick their tongue into the insect tunnel in the tree. Now, I'm going to tell you something weird here. Some woodpeckers can extend their tongue 3 to 5 times the length of their beak. Don't believe me, huh? Now, that's a short one. That's about 3 times the length of the beak. I'm going to go 5. Now, you got to ask yourself a question here if you're an evolutionist. How by small, steady steps, mutation, natural selection, do I go from, say, have you ever seen a bird's tongue? You ever have a bird at home, like a parakeet or something? I mean, it's a little tiny thing, right? If you've seen a mockingbird or a robin in the Midwest, sometimes their tongue is visible, but it never extends the full length of the beak. The muscle is a tongue. I mean, the tongue is a muscle. I've been up here a while, huh? Tongue is a muscle that attaches itself to the lower jaw. And, you know, you can do something. I mean, we have some strange humans who can touch their nose with their tongue, and, you know, that's a genetic mutation, by the way. It's not a harmful one. Okay, so I have one of those thumbs that bends back almost at 90 degrees. Okay, that's a rare trait, too. Most people can only do that, but I can 90 degrees. All right, that's a weird one. So I'm weird, too, just like you. But in the woodpecker, the tongue, the anatomy of the tongue is very, very different. Because you ask yourself the question, that's a good thing to have, a tongue that can really extend, go way out, okay? It's how it eats, how it feeds, how it survives. What does it do with the tongue when it comes back inside the head? Where does it go? Well, let me think about that. I mean, that's kind of weird, you know? What does it do, just roll it up or something, you know? And no, it doesn't do that at all. Let me show you. Now, we're going to take a little journey here. I'm going to use some red arrows here. We're going to start at the tip of the tongue of a woodpecker, and we're going to follow it back into the throat and into the head. The tongue of the woodpecker does not attach to the lower jaw. It's like no other vertebrate around. We'll start at the tip of the tongue. We go back into the beak. It makes a loop down into the throat, loops around to the back of the throat, comes up between the skin and the skull, behind the skull, up to the top of the skull, under the skin, and above the actual skeleton itself. Now look to the other photo on the right. Comes down between the eyes, inserts into the right nostril, and extends all the way to the tip of the upper portion of the inside of the upper portion of the bill. When it wants to extend its tongue, it loosens the attachment there inside the upper portion of the bill and reattaches between the eyes underneath the skin. And those big loops behind the head, the big loop in the throat, all flatten out, and the tongue is a muscle, so it contracts, and it shoots out the mouth 3 to 5 times the length of the beak. Now, if you're going to evolve a woodpecker tongue, you've got 2 problems to solve at the same time. Number 1, you've got to evolve a longer tongue. In some respects, I would say that's no really big deal. You get a mutation, and maybe the tongue comes out another, half a centimeter. But the first time you go to drill a hole, you cut it off, and that's not going to help you. Why? Well, because you also must evolve a retraction mechanism. If it's just a tongue that's already attached to the lower jaw, a longer tongue isn't going to do you any good. There's no place to put it. So you also have to have a way to pull it back inside the head. So you have to do 2 things at once, a longer tongue and a retraction mechanism. Anytime evolution has to try to do 2 things at once, it fails. It just utterly fails. It can't do 2 things at once. Oh, I love Stephen Jay Gould. Too bad I know he's passed away, but he always gave us some great quotes. He was talking about these kinds of transitions, and he said, but how can a series of reasonable intermediates be constructed? The dung-mimicking insect is well protected, but can there be any edge in looking only 5% like a turd? Sorry for the kids here, but they like it too. Now, I'm going to have to ask you to think about this a little bit. The toughest stage for evolution is always these incipient stages towards an adaptation. And Gould has put a beam on the problem. We have insects today that look like a bird dropping. And that's why no other birds eat it. They are well camouflaged. If you come across one, ooh, what's that? Oh, it's moving. You know what? It looks like a turd, okay? And it's very effective. It's very well camouflaged. But I ask you, as Stephen Gould does, what does only 5% like a turd look like? Is that going to deter a bird from eating it? Because it looks 95% like what? A real insect, something I eat all the time. So how is that incipient 5% stage going to do that insect any good at all? How is that going to be selected for? Why would it be selected to get better and better? If you've got an insect that starts off looking 90% like a turd, I could understand how selection might make it just a little bit better. But it's these early stages that it can't get past. It can't accomplish the task. It's not equipped to do that. Now I'm going to show you something here. This is Big Dog, and this is done by Boston Dynamics. This is on a defense contract for Boston Dynamics. It was done it was in process about 4 or 5 years ago. This film clip is from about 3 or 4 years ago. And I got this from Paul Nelson. Some of you have seen Paul Nelson, some of the other Illustra Media films. And his brother was involved in this research project. Big Dog was a government contract to come up with a mechanical mule to pack supplies for the Army into rigorous terrain. Okay? So you're going to get a lesson here on what Big Dog can do. Oops. I got to do the same thing. Excuse me, 1 second. Each limb, each joint communicates with the mainframe computer 500 times a second. There are 3 slips. not exactly an Olympic long jumper, but now, is there any question in your mind that Big Dog is designed? Nah, you got that pretty well figured out. You intuitively understood that. Now, let me throw one more curve to the engineers designing Big Dog. What we also need Big Dog to do, we need two different types of Big Dog. And in one of those, we need a small capsule by which when the two of them get together, they will form a new tiny Big Dog that can grow on its own. Right? What's the engineer going to tell you? Go away. You're crazy. We can't do that. But yet, I've got a Big Dog at home. I've got a big Irish setter. And, you know, and I've got a golden retriever. You put the golden retriever out there in the field, you know, if he wasn't fixed, he wouldn't have any trouble knowing what to do. Right? Now, and the nose of a dog is far more sensitive than anything they'll be able to build into Big Dog. And yeah, Big Dog's got some interesting capabilities here. I mean, the slips and the falls, and I'm not sure, you know, my Irish setter is 13 years old, and there's no way he could handle that. Okay, he'd have been flat on his face. All right? But at the same time, a real dog, a biological dog, does so many more things than Big Dog could ever be designed by humans to do. But yet we're so convinced Big Dog is designed, but we want to say the biological dog has evolved. What we're seeing is that as we investigate more about what design means, as we investigate more how the genome operates, as we investigate more how populations exist in nature, we continue to see additional limits to biological change. Yes, we do see change, but we're finding more and more that there are far greater limits to change than there are opportunities for biological organisms to change on their own. Sternberg also did another presentation this last year. He called it Why We're Not Chimps. And now that they have both the human genome and the chimpanzee genome fully sequenced, and now they're comparing the two. And yes, the structural genes are indeed 98% similar. But as I just showed you, the structural genes have little to do with what separates one organism from another. Where they really differ, and they differ by orders of magnitude, he's saying, is in the genomic architecture outside of the protein coding regions. They are vastly, vastly different, especially for those that have anything to do with brain function. The structural, the organization, the regulatory sequences, the hierarchy to how things are organized and used are vastly different between a chimpanzee and a human being in their genomes. So that 98% that's supposedly junk is nothing but. It's astounding today. We're coming up with more and more observations. And what I really loved is 2 years ago, I asked Sternberg at the end of his immaterial gene presentation, I asked him, I said, well, you know, okay, all these articles you've given are in the mainstream literature. So all of these genomic researchers must be aware of this. Some of them are evolutionary genomicists, and they're studying the evolution of genomes. What are these people saying about what you're finding? He says, well, it's a good question. I was wondering the same thing, and I still have a few friends up at the National Institutes of Health out in Washington, D.C., so I called a few of them, and all they told me was that they're not talking. They're not discussing the evolutionary implications of what they're finding, of the different types of animal genomes. That says volumes to me. Because what I hear is that you're coming up with information that they have no way to interpret. They have no way to understand. They can't explain it. They can't understand how it got to be that way in a purely evolutionary naturalistic process. So I am very encouraged today. I'm very encouraged, and the more we know and the more we dig into the genomes, how it functions, how it operates. I gave the immaterial genome presentation to a group of Christian school teachers who were science teachers, and I remember one guy telling me, you know, he raised his hand and said, you know, I just finished a genetics course at the University of Kansas, and they didn't tell us any of this stuff. And another guy said, this changes everything. It literally changes everything, what we're learning today about what genes are, how they operate, how they function. And so And everything we are learning is coming our direction. The stuff we're learning ain't going the other direction at all. So this is an exciting time, and I wish I was almost 30 years younger so I could go into research. That wasn't really an option for me 30 years ago when I did finish, or 20 years ago, but I'm glad I'm here today. Questions? Yes, sir.
Two questions. Two questions. What is the organism, or what organism has the fewest number of genes, and what is that number?
The smallest number, well, if you're talking about a bacterium, those have about 600 genes. Now, there are smaller bacteria that they're estimating they found about 400, 450 genes, and they're trying to guess what the smallest genome could possibly be.
Okay.
And the smallest estimate they come up with is usually about 200 to 300 genes.
All right, and we have tons and tons of single-celled animals.
Yeah.
And then we have ones like us with trillions. Are there 2-celled, 3-celled, 4-celled, 5-celled, 6-celled? How does it go from 1 cell up to?
Oh, those have all gone extinct. Back in the.
But what do we actually find?
Well, from let's see, we go from single-celled protozoa, which would be amoeba and things like that. And then you get into some that are a little bit bigger, just still single cell. And then you get some that are aggregates, and they're still individual cells, but they aggregate together. They don't seem to have much in the way of cooperation. Those can be 10 to 100 cells. But when you really talk about a functioning organism that has more than one type of cell, you're talking about a sponge, and that you can have still hundreds, thousands, tens of thousands of cells. So we don't really have organisms that function with, say, 2 different types of cells, but there's only 5 total. We don't have anything like that. Go ahead, just whoever you can get to there.
Get one of you guys first,
okay. I apologize, I'm going back to before you started talking about the main point of your whole talk, to where you were talking about the difficulty of people becoming interested enough in the subject to even come and listen to it.
Yeah.
My observation has been that we have this juggernaut that convinces people not to even listen to anything that has to do with Christianity or biblical truth or truth at all, for that matter, with anti-science being promulgated through the schools, and the national media has like a death grip on information. Your typical person comes into society as an adult completely indoctrinated and completely unwilling to even listen to things like you're talking about today. Of course, that ties back to what you were saying, is that you're changing the direction of the ministry.
Yeah.
But you didn't really get into how you're changing it and how you would expect to be able to reach a generation that's just brain dead practically. How do you? What directions are you exploring to do that? Is the question.
Well, it's a good question, and I will try to be as complete yet brief as I can. At the Probe website, which is probe.org, and if you just go to vision/vision, we do have some information there, but we haven't put a lot on the website yet because we're still developing a lot of this. The first thing is we've just recently contracted with the Barner Group to do a special survey for us of 18 to 40-year-olds, 800 of them, those who claim to be born again, and who either go to church or don't go to church, but find out from them what authorities do they listen to, what kinds of medias do they pay attention to, where do they get their information, who do they trust, who don't they trust. If you go to church, why? If you don't go to church, why? We need to get a better understanding ourselves of how these young people think, what they pay attention to, what they don't. They're not going to come listen to a lecture like this. We already know that. Okay? So we're going to have to come up with different ways. We don't even know what some of these possibilities are. One of the researchers that we heard from recently at a National Religious Broadcasters Research Symposium indicates that this younger generation, especially 18 to 30-year-olds, are all over the map as to what sort of media they use. They don't really watch much TV. They don't really listen to Christian radio. It's all through the iPods. It's all through their mobile phones. It's through Facebook. It's social networking things, opportunities. And so we're not even sure at this time what kind of approach we're going to use. But here's the basic idea. We've broken it down into two stages. One of them we're calling breaking free. Somehow we're going to need to construct a church-wide as well as an internet web-based, I hate to even use the word curriculum, but program, whatever, that forces people into situations where they have to stop and think, what is it I really believe? Why do I act this way? And why did I come to these decisions? And here's what the Bible says. Nancy Pearcey has a brand new book out. She wrote her book, Total Truth, in 2004, which I highly, highly recommend that book. Her second book now is called Saving Leonardo. And in the opening chapter, she refers to a couple of examples which specifically highlight the problem we're dealing with. She said in the 2008 election, there was a ABC News reporter who went to a Christian youth rally in New York City. So she's talking to Christian youth. And she says in the paragraph that you'd be encouraged that the vast majority of these youth, meaning high school kids, had a very pro-life stance on abortion and life issues. But the reporter found out that all the candidates they were supporting were all pro-choice. And the reporter says, now this doesn't seem right to me. Doesn't this bother you that you say you believe this, but you're voting for these people who don't believe that? And the response was just amazing. They said, well, yeah, it bothers me a little bit, but after all, it's just personal preference. And you can't hold somebody's personal preference against them. I just thought, oh my gosh. How you look at life, how you define human life is a personal preference. That is how they have been intimidated by the culture, and to what degree. So there's a dissonance there. And those are the kind of things that we're calling this breaking free, as I said, and we're going to have to find ways to break through that dissonance and allow them to see. For some, it might be in bioethics, like with pro-life. For some, it might be how they use and steal songs and things and trade back and forth, and they violate the law left and right. They all do it. And somewhere, somehow, there's got to be a connector. And we're not quite sure what those are just yet. If we get them through that point of helping them to understand that they have been taken captive by the culture, and there is far more to their Christian life and worldview than just having a ticket to heaven. Because some of them will actually tell you that I love Jesus, I read the Bible, and I'm so glad that I have trusted in Him, but if somebody else over there finds a way to heaven through Hinduism, that's okay. They found their way and I found mine. And these are people you're going to find in our churches, in our youth groups. But we're not asking them those questions all too often. So there is a huge, huge disconnect. And we are banking everything at Probe Ministries at trying to find a way to break through that cultural captivity. We've got about a 2 to 3 year research and development process ahead of us. And it's going to take money we don't have. We are going to all kinds of new different kinds of sources to raise the funds to do this. This is going to cost us 5 to 10 times our normal operating budget to achieve it. But if we have a church-based campaign that, for those who do attend church, and it's done something like 40 days of purpose, where the whole church did it at the same time. If you just do a Sunday school class that is breaking free, ain't nobody going to go. We already know that. It's got to be something that goes throughout the entire church. Or somehow through the social networking concept, that's how the internet and web-based product would be used. Because word develops. they develop associations not from the top down. In other words, if the authority of the pastor tells them this is something you ought to do, they're not likely to listen. But if their friends have discovered that something is good, something's worthwhile, you ought to see this video, go see that, go do that, then they'll go look at it. They'll go pay attention to it. So there's going to have to be both the top-down approach and the bottom-up approach. And I'll be honest with you, we're scared to death. There is a lot for the church in America writing on this problem. I'm not saying we're going to solve the problem for the church in America. But our new vision statement is quite simple and scary. We want to free the minds of 50 million culturally captive Christians around the world and build them into confident ambassadors for Christ by 2020. We have to have 10 years. We have to. In the first 5 years, we're going to focus here on the States. After that, we're going to go international. We have a number of international connections to try to do that. But this is going to be an all-consuming effort for our small staff. We're going to have to get lots of outside help. We're going to have to hire outside people to do a lot of this, even the writing of it. I can't write to 18 and 40-year-olds anymore. I can't do it. I'm 57. I don't really know how to communicate to them anymore. I have trouble communicating to my 30 and 28-year-old sons. And they're in the target group, okay? I've known them all their lives. So I can't answer you directly. We have to get this research from the Barna Group done. We'll have this back to us sometime in November, hopefully early November. And then we begin to bring other creative groups and voices together and say, okay, how do we put something together to reach these people? We already know what they believe. We already know how they live their lives. And they are extremely captivated by tolerance and relativism. Yeah, a third of them, I say over half of those 18 to 23-year-olds would say that the Bible is inspired by God. I think 70% would say the Bible is inspired by God. But only about half of those would say that the Bible is accurate. Now think about the dissonance with that. What does that say about God? He inspired it, but he couldn't keep it that way? Or he inspired it, but he didn't get it right in the first place? Or God isn't all-knowing? God isn't all-powerful? Who is this God who could give you the Bible? It's inspired, but it's not accurate. There are some real issues and problems here, some real dissonance going on, and just exactly what it's going to take to break through all that, we've got a lot of uncovering to do. Yeah.
Okay, Ray, we have another
Yeah.
question.